Air pollution refers to the contamination of the air in our environment by harmful substances, often the result of mankind's careless exploitation of nature's resources. As humankind's agricultural technology increases, less individuals and systems need to be devoted to the production of food and necessary items for our survival. The result is an increase in the amount of stuff that we are capable of creating. While this kind of increased production capability causes a very welcome increase in our standard of living by relying on such agricultural technologies, which are among the most non-renewable fuel intensive, we've created a way of life that is ecologically unsustainable. The experiments performed and presented by our group attempt to illustrate the harmful effects that acidic pollutants have on plant life. Acids have harmful effects on plants because they denature essential enzymes. A hydrogen ion dissociates from sulfuric acid. The hydrogen ion then bonds with carboxyl amine and R groups of amino acids. This may disrupt the existing hydrogen bonds and disulfide bridges between amino acids or the hydrophobic van der Waals interactions between nonpolar R groups and alter the secondary alpha helix and beta pleated sheet and tertiary structures, how the protein folds. As a result, the protein loses its function. For example, an enzyme has a specific shape where it catalyzes a reaction by correctly orienting substrates in an induced fit active site. It lowers the activation energy required. However, when an enzyme is denatured, there is no catalyst and there will be less particles with enough energy at the correct orientation. This is represented by a leftward shift of the maximal Boltzmann distribution curve. As a result, the cells of a spore may not be able to catalyze some of the reactions necessary for its survival. The purpose of this lab was to investigate the effects of acidic environments of different pH on germinated and non-germinated clover plants. The independent variables were acidity of environment and maturity of the plant, germinated or non-germinated. The dependent variable was plant growth and was measured by height in millimeters. Control variables were plant species, plant age relative to those of the same type, for example, all germinated and all non-germinated, amount of water given, and environment with the exception of acidity. Given that plants naturally live on rainwater with a pH of between 5 and 6, the group predicts that adding strong acids with pHs of 1 to 2 into a plant's growing environment will have adverse effects on the plant's development. This is because strong acids denature enzymes by disrupting the hydrogen bonding and IMFs between R groups. In our case, the H+ is expected to dissociate from the H2SO4 and bond with the R group of important enzymes, denaturing them and having a negative impact on the plant.
Materials: a digital camera, 3 pipettes of 10 milliliter capacity, 3 volumetric flasks of 1000 milliliter capacity, 50 milliliters of 1 molarity H2SO4 sulfuric acid, 16 petri dishes, distilled water 3000 milliliters, 16 filter paper, scissors, and 160 clover seeds. Procedure: we first got graph paper and drew circles on it so that we could add this graph paper into the petri dishes along with the filter paper, preventing any leakage of solution of acid, which could ultimately affect our data. We then cut the filter paper and graph paper with clean scissors so that it would fit inside the petri dishes. To prepare the different solutions of acid, we got the 3 volumetric flasks and put in the amount of sulfuric acid needed to produce the desired molarity of acidic solution. In the 0.02 molarity product, we put in 20 milliliters of acid and 980 milliliters of water. In the 0.01 molar product, we put in 10 milliliters of acid and 990 milliliters of water, and followed the same procedure with the 0.005 molar product. After putting in different volumes of sulfuric acid into the volumetric flasks, we filled the remaining volume of flask with distilled water, which you can see Brendan doing here. After getting all 3 volumetric flasks full, we shook them to make sure that the acid was equally distributed inside the flask. We then poured this solution into containers which we could take home. After getting all of our equipment ready, we proceeded to divide the 160 clover seeds into 16 petri dishes. We then added the different stocks of solution to 4 petri dishes each, and the remaining 4 petri dishes were grown by using distilled water, our controlled experiment. We then recorded our observations of the growth of beans over a course of 10 days.
Here we have the germinated trials on the left side and the non-germinated trials on the right side. We've arranged our petri dishes in increasing molarity from left to right.
On day 1, many of the seeds have germinated, especially for those which are more acidic and those which are in the non-germinated groups. The radicle also appears to be forming. On day 3, the plants are continuing to grow and there are more leaves forming. There seems to be brown spores forming on some of the cultures for the germination trials and some of our control trials. On day 5, there are many leaves in the acidic cultures and some in the neutral medians. The plants are developing a taproot, suggesting that the clover leaves are dicot angiosperms.
On day 10, all the plants have desiccated. The paper is dry and the plants are extremely white and thin. There's a high proportion of spores on the neutral and germinated trials. From this lab, we observed that an acidic environment is beneficial to plant growth because it helps kill spores. But in the long run, the acidic environment is harmful to the plant after it develops its root system. Hence, we observed that the plants that grew best were the ones in the 0.005 molar of acid, because it was strong enough to kill spores, yet it was not as harmful to the plants as in the more acidic environment.